All right, in this video, we're going to talk a little bit about the cell cycle. So basically, the cell cycle is kind of like the life cycle of the cell. Once the cell is formed, obviously all cells come from division of other cells through mitosis. It's going to grow, it's going to mature, it's going to divide again. So the cell cycle is basically divided into 3 to 4 steps. So G1, S, G2, those are all part of interphase. That's when the cell is doing what the cell does. And then there's mitosis, when the nucleus divides, and then there's cytokinesis also. So all the cells in our body come from somewhere else. So epithelial cells, blood cells, stem cells, most of the cells in our body are going to follow this particular pattern. But some cells, once they form, they grow, they mature, but then they don't divide. They go into the G0 or the resting phase. These include things like brain cells, nerve cells, muscle cells. We used to think that a lot of these cells couldn't divide, but what we found out is that something is stopping them from dividing in many cases. Sometimes cells can go into the resting phase for a period of time and then come back out, like the liver cells. So let's talk about the steps of the cell cycle briefly. So basically we have interphase, which is the time between cell divisions. 90% typically of the cell's life cycle is going to be spent during interphase. Now again, cells are going to spend different amounts of time in these different phases depending upon the kind of cell we're talking about. So this is basically the cell's doing whatever that cell is supposed to do. It's making RNA, it's making proteins, enzymes, doing those things that the cell is supposed to do. It's also the time when it's getting ready to divide if division is called for. And that's mitosis and cytokinesis. So during interphase, the nucleus is well defined. When we talk about looking at a typical cell and looking at the nucleus, this is the type of nucleus we're talking about. The DNA is loosely packed in long chromatin fibers. It's an uncondensed state. You can't really see it with a light microscope. You could with an electron microscope. The cell does prepare for mitosis. It's going to copy the chromosomes during one part of interphase. It's going to make proteins that are needed for cell division. It's going to make organelles so that when the cell divides, each cell has the right number of organelles and so on. So we divide interphase into 3 steps. There's the G1, the first gap phase or growth phase. This is typically the cell doing its normal thing. Sometimes cells will go from G1 into G0 like the nerve cells. As the cell grows, maybe it receives a signal saying, hey, we need to divide. Okay. Then the cells would move into the S phase, or the DNA synthesis phase. This is when the DNA, the chromosomes, get copied. From there, the cell would move into the G2 phase, the second gap or growth phase. This is where the cell is going to do all the preparation it needs for division. It's going to grow some more. It's going to make organelles, proteins, it's going to make membranes. It's going to do all the things it needs to do to make a new cell. So during the S phase, this is when DNA replication happens. It makes an identical copy of its DNA. So now you have a copy for each new cell. So when the human cell duplicates DNA, it's got to copy 3 meters of DNA. And it's got to do it exactly right, or it can cause a problem. So the error rate for humans is about 1 mistake per 100 million bases. That seems pretty good, but there's 3 billion bases in a typical mammalian genome. So that's, still a significant number of errors. So our cells actually have mechanisms for making repairs to mistakes made in copying the DNA. So again, in a typical human genome, error rate of 1 per 100 million, that's 30 errors per celled cycle. That can lead to mutations. If it's in the body cell, it can cause problems like cancer. So to keep the DNA organized during mitosis, one of the things that has to happen is the DNA has to get organized into the chromosomes. So we start with a double helix. Okay, there's our DNA. The DNA wraps around a special group of proteins that form like a spool called they're called histone proteins. So that kind of coils up the DNA. And it kind of looks like threads on a spool. This is what we typically call chromatin. Okay. Long, thin fibers. Okay. In mitosis, the chromatin condenses further into the chromosomes. Okay. So after DNA duplicates, the chromatin is going to condense. It's going to fold, it's going to coil up, it's going to form into, going to get these supercoils and all these different things. And it's going to form a typical chromosome. In order to help the chromosome form, help organize it, we have scaffolding proteins that form chromatin loops and so on, all these different organizational structures that lead to the formation of the chromosome. So here's an electron micrograph of what a double-stranded human chromosome looks like. So when you duplicate the chromosomes, you're making a copy of each chromosome. Remember, one came from the dad, one came from mom. So you have, again, so you can see the process. And then once the cell's ready to divide, that duplicated chromosome is going to separate into two different cells. Each cell has the same number of chromosomes. So that duplicated chromosome is made up of two sister chromatids joined together at the centromere. So we call these homologous chromosomes. Homologous simply means they have the same information. Single-stranded, double-stranded homologous chromosomes made up of sister chromatids. And then mitosis is basically the time when the DNA is being divided into two daughter nuclei. So it's division of the nucleus. There's 4 phases. You guys probably learned these before: prophase, metaphase, anaphase, and telophase. In a future video, we'll talk more about each of the 4 phases. Then typically mitosis will be followed by cytokinesis, the division